I talked about a very interesting discovery wherein it could be possible to culture organs of reproductive in nature like infant and the ovule, how that culture revolutionized the crop improvement programs world over. And fortunately, this discovery was made in India, which was later refined and refined and utilized for crop improvement globally, including in India. Now, if we look at the era of 50s and 60s for tissue culture uh, was one of the unique era, I would say, that a lot of discoveries were made during this period. And one such discovery which I'm going to discuss with you is related to single cell culture. From explants to callus, and callus, when it undergoes, you can tease it in the liquid media so that all the cells get separated, and it could be possible to not only taking callus and differentiating it into plantlet, but it was possible if you take cell suspension cultures or single cell or group of cells and culture them, then what they do, they first form the callus, and then that callus undergoes uh, differentiation and plantlet formation. But main problem, it was seen that plant cell has a covering, what we call cell wall, which animal cells don't have. So during this period, what was seen that animal cells could be cultured and utilized for different purposes. But but plant cells, though they were cultured, but because of the cell wall coating, it was not possible to further manipulate these cells. The idea or the concept which was thrown was that when it was demonstrated that a single cell can give rise to a plantlet. So the concept was that if one can genetically modify this cell, then from this genetically modified cell, you can make a, you can genetically modify a plant in vitro. But what was realized that because of the presence of cell wall, that manipulation was problematic. So therefore, the plant cells were also made to see that if they can if one can get rid of the cell wall, and the plant cell also become like the animal cell, means cell without cell wall, only having the cell membrane covering. Then, like animal cell, plant cell also can be manipulated. So, a whole lot of work went in to achieve this. That is, first they get rid of the cell wall. Then you have a cell without cell wall, what we call, I mean, the name was given as protoplast. And then this protoplast was made to regenerate. And when protoplast regenerates, it first forms the cell wall. So from protoplast, it becomes cell. And then this cell then follow the normal regeneration process to make plant. So all this uh, process was developed in 60s. Okay, and this is what I'm going to talk about in this session. How the protoplasts were made, and how then they were made to regenerate and give a give a plant which is generated or which has been generated from the protoplast. Okay, so I'm talking about this. So let us begin. Begin looking at what is this protoplast. As I mentioned, protoplasts are plant cells where the cell wall has been removed. Okay, and when you remove cell wall from plant cell, it becomes protoplast, and that's the name given to it. A naked cell or a cell without cell wall, we call it as protoplast. And when you view it under microscope, they look like this, very shiny cells, you know. And when you culture these protoplasts, you can give rise to, in 5 weeks, you can have the regeneration into plantlet formation. So this is nutshell which was achieved. Okay. Now, how to isolate protoplasts? Now, this is again an area which was very, actively worked upon in early 60s. And the first attempts were physically remove cell wall from the protoplast, what we call mechanical methods. And subsequently, when the chemistry or the biochemistry and the enzymes means what constitutes the cell wall, that is, the complex carbohydrates like cellulose, pectinase, and when it was discovered that there are enzymes within the plant cells, which plants make, which can degrade these complex carbohydrates like cellulase and pectinase. So that is, I'll come to that later, but then what I'm trying to say is that in absence of the second discoveries of the cell wall degrading enzymes, first attempts were made just by using mechanical methods to remove physically cell wall from cells. And how that is done, I mean that is a very tedious process to individually look at the cell and then cutting the cell wall with a knife and release the protoplast and collect the protoplast and then regenerate. And a lot of reports are there where it was successfully done using mechanical methods of removing cell wall by knife. You know. But then it is very cumbersome process as against the use of enzymes. As I mentioned to you, these complex carbohydrates which constitute cell wall like cellulose and pectinase. So the procedures which were dubbed up with the single step and the two-step processes. Single step means you can use both enzyme as a cocktail of cellulose and pectinase, like this here. Use that, and then that protoplasts are released in a couple of hours. What you have to do is take the tissue, tease it a little bit, and then incubate it into this cocktail of enzymes. And then after 3-4 hours, the protoplasts are released, and you can purify these protoplasts. And in the second protocol, which was two-step protocol. What is this two-step protocol? First, you use the pectinase, okay? So release of isolated cells, and then you use the cellulase to degrade the cellulase. So that is two-step. First, use of pectinase, and then use of cellulase, and ultimately both result into release of protoplasts. So again, it was from two-step process to one-step process. And today, it is very easy to isolate protoplasts from any source. You take any source, tissue, leaves, stems, roots, and just incubate these tissues into this cocktail of enzymes. and you will be able to release the protoplast. Then purify, you can filter them, and you will have very nice preparation of protoplast in hand. And these protoplasts in hand, can be used for a variety of purposes. I will talk about that later. But then let us look at what all it involves. So once you have isolated the protoplasts, they need to be put on an appropriate medium to reform the cell wall. You have removed the cell wall, and then now, after whatever manipulation one need to do, this protoplast need to undergo regeneration. When it is regenerating, what it does, it undergoes the cell wall formation as the first step. And from one cell to many cells, and then callus formation, and then regeneration by organogenesis or somatic embryogenesis. Okay. These are the two routes always available for plant-like formation. So, like any other culture, this also needs to be optimized. That is, there are so many factors which I have listed here. That is the density of the protoplast to begin with, how many protoplasts should be taken, and then the growth regulator concentrations need to be optimized, and temperatures need to be the pH and the growth regulator. So all these factors need to be optimized, for successfully culturing protoplast. So continuing on that, when you do the protoplast culture, nutrient media needs to be optimized, and first the cell wall formation, and then this cell divides into two and couple of clumps of cells, and then small micro colonies like the bacterial colonies you have seen or you will be seeing in the practicals. Similarly here, the single protoplast undergoes cell wall formation and the division, and this division accumulates clumps of cells, and then the colonies can be seen on plate. And then from clumps, you can generate the whole plant, as I mentioned, through organogenesis or somatic embryogenesis. So this is summary of the events which take place from plant to the protoplast isolation, purification, and then you have the plating of this year. The single protoplast divided into 2, 4 clumps are formed, callus formation, and then shoot regeneration, and the root regeneration. So this is the basic technique, basically. So, as I mentioned, the idea was to have a cell which is comparable to animal cell, and animal cell is the one which is without cell wall, and plant cell had a issue of cell wall coating outside, which was a limiting factor in terms of manipulating the cell, because the cell wall doesn't allow anything to get in. So that with that idea in mind, this whole technology was developed, whereby you can have the naked cell generated or the plus generated, and then optimizing conditions for regeneration of these protoplasts into whole plant. So now the platform was ready, wherein the manipulations can be done at the single cell level, at the protoplast level, in terms of genetically modifying. So that I'm talking about in 60s. And at that time, there was nothing known, linkage between trait and the genes. It was not known. But what was conceptualized, that if we can have a plant which can fix atmospheric nitrogen, taking genes, and as you know, there are plants like cereals, they don't fix nitrogen, and there are legumes which can fix nitrogen through a mechanism of through symbiosis of rhizobium, for example. So on. So idea was that if the cereals can be made to fix atmospheric nitrogen by genetically modifying them with the genes which are responsible for nitrogen fixation in legume plants. That was the concept thrown in 60s itself, but that was actually the the farsightedness of the scientists of that time, you know. But then, I mean, this could not be realized even today. We have not able to make cereal plants to fix atmospheric nitrogen, but then it gave it infuses the enthusiasm among the scientists of that time to start working and to understand how the nitrogen is fixed to begin with in the legume plants, what is responsible, how this symbiotic relationship between rhizobium and legume plant is established, and the bacteria resides in nodules and nodule formation and all that. And that gave rise to a clear-cut understanding about this process. But then that this could not be mimicked in cereals, you know, as I mentioned. But then, leaving this story behind, uh, uh, by successfully demonstrating this phenomenon of protoplast isolation and culture and raising plants from protoplast was the kind of research done in 60s and 70s. From economically important plants, this process was demonstrated. What is this process? I'm referring to isolation of protoplast and regeneration of plants from protoplast, with the idea to keep the technology in place so that someday, if we have the genes, we can manipulate this protoplast. And if we also have the methods of sending the foreign gene into the protoplast, then it would be possible to make genetically modified plants, or what we call transgenics. So that was the idea. So with this, a lot of work was done in 60s and 70s to develop protocols for isolating protoplasts and regenerating them to the whole plant plants. And as I mentioned, I'm not going into the details of this. These are the factors which affect chloroplast culture, and these are the general factors which I had been talking about, which are true for all sorts of in vitro manipulations. Like, for example, plant species and the varieties, the source from where you are taking, they are the very, very critical factors. Here also, age and which organ you are taking, you know, it is seen that the mature tissues respond differently as compared to the young and softer tissues. So, and then the pre-culture conditions also is another factor which affects, and then the pre-treatments of the tissues before you isolate protoplasts, and density has to be optimized. How many protoplasts should be plated on the culture plate? If you take too many protoplasts, it is counterproductive. If you take too less protoplasts to begin with, it's also counterproductive. So all these have to be optimized. And as I mentioned, what are the importance, or what is the importance of this protoplast culture technique as a whole, you know? And there's so many, pluses which is seen, and so many phenomena which were studied using using chloroplasts. The osmotic behavior of the cell could be studied, because the cell is naked, and it will react to the osmotic stresses of the cell where it is exposed to. Hormone applications, how cell respond to hormones, that is could be seen. And then the cell wall formation. When the protoplast undergoes regeneration, it regenerates the cell wall. So how the cell wall is formed, process was studied, and then organogenesis, morphogenesis, and very importantly, the gene transfer. A lot of work was done to study the gene transfer, that how genes can be transferred to the to the naked cell or to the protoplast. And in 70s, lot of efforts were made to transfer foreign DNA into protoplast using different approaches. The naked DNA was used, the protective DNA in the form of liposomes was used, but all these attempts which were made in late 70s and early 80s did not give the transformation. They are all, if you scan through the literature, you will see n number of papers where the methods were tried to transform this protoplast, you know. And what was realized later, once it could be possible to transform plant cells or protoplast, is that at that time, what was missing was the selection process. When you have the say population of protoplast and you are sending the DNA, so you have to from this group of cells or protoplast, you should be able to select the one which is undergone transformation, and that process was missing. What I'm referring to the selectable markers. In mid-80s, that deficiency was overcome by the discoveries of antibiotic resistance genes, in bacterial systems. And then once they were discovered, then these genes were used as the selectable markers to begin with. So it was very easy after transformation process, you can select the transformed cell in presence of antibiotic, for which you have tried to push the gene to give resistance. So what is done simply, you take the culture media and expose the cells to the antibiotic. Any cell which has undergone the transformation with that gene will survive and others will die. And that was the one breakthrough in mid-80s, which could be possible to demonstrate genetic transformation. And I will talk about that later, that story. So this was one of the factors in 70s, and when efforts were made to develop technologies to protoplast transformation, that they could demonstrate the entry of DNA into the cell, into the nucleus, but then they could not select a transformed protoplast or cell because the absence of any reliable selectable marker. So these are some of the importance of this protoplast culture, that a lot of phenomena were observed, studied, and details are known today. Another thing which was observed while culturing the protoplast, that these protoplasts tend to fuse with each other. Okay, so they undergo hybridization. Okay, so it can, and what was seen and gave rise to very interesting possibilities, which was tested in the 70s. I'm coming on to the somatic cell hybridization, those kind of possibilities. Because when it was observed that the protoplasts can fuse spontaneously if they are left in the media, then it was thought that it is possible to raise hybrids. Hybrid how? If you take two protoplasts from two different sources and fuse them, then you have a single cell or hybrid cell, and this hybrid cell can undergo regeneration and can give you a hybrid plantlet, which has its root from two different somatic cells. The hybrid cell produced by the hybridization of two cells from different sources. Now, these two different sources can be two different species of the same genus or two different genus. So it opened up a different area of research, wherein normally the sex cell fuses within the same species, plants from the same species, and that's how the sexual reproduction goes. So this somatic cell hybridization opened up a possibility wherein you can bring two different genetic makeup together in a hybrid cell and generate a plant form. So we will see some interesting, discoveries in this. So, okay, what is a somatic hybrid? So it is the develop, as I mentioned, and I'm just trying to defer for you that it is development of hybrid plants through the fusion of somatic protoplasts of two different plant species varieties are called somatic hybrids, basically. Okay, which is different than the sexual reproduction, as I mentioned, where the sex cell from the same species can fuse and reproduce and give rise to the new plant. Here, these two somatic cells taken from two different sources, from different varieties, species, genera, and then theoretically it should be possible to get the hybrid plant. But what actually happened? That is. So basically what happens in somatic hybrid, it involves two somatic cells and then tissue culture is essential. And here, when this hybrid cell is produced, it has the equal contribution of the two cytoplasms. And when it happens, it is overcoming the barrier of cross incompatibility. Means when you can bring two somatic cells from two different sources, we are overcoming the sexual incompatibility. As I told you, the sex cells can fuse only within the same species. So, but by fusing the somatic cells from two different sources, we are overcoming this barrier. And that's what. And that means that you can have a novel combination of genes of unrelated species coming together or getting formed, which naturally is not possible to bring two gene pools together by sexual means. It could be, it was thought that it is possible to bring two different gene pools together to get newer combinations by somatic cell hybridization. So you create a somatic cell hybrid, and regenerate a platform here. So that was something very, very exciting in the early 70s when the protocols were isolated, regenerated, and this was seen that, okay, it can be undergo spontaneous fusion. Okay, so how they fuse, as I mentioned, that there are two ways: spontaneous fusion and induced fusion. You can induce the fusion. So this spontaneous fusion can take place intraspecific and intergeneric. Intraspecific and intrageneric. Okay, so two different genera can protocol from two different genus can undergo spontaneous fusion, and the induced fusion can be through chemicals, mechanical, or electrofusion. These are three techniques which have been successfully used and demonstrated. Either by chemical means or mechanical means or electrofusion. So why one need to induce this process of fusion? Because spontaneous fusion frequency is pretty low. And if you want to fuse in large numbers of the fluid and then try for regeneration of the hybrid cell into whole plant, then it is the number game. So you require large number of cells participating in fusion and then selection. I mean, then the of course the selection part will come to that, because here when you mix two different cell types, here in this group you will have hybrid cells and the parental cells also exist. So you have to pick up your hybrid cells effectively and then regenerate. So this selection is required here also. Selection of the hybrid cell among the population of parental cell A, parental cell B, and then the hybrid cell A and B, which is the product of A and B fusion. Okay. So, but then methods are available and have been used successfully, chemical electrofusion, which induces the frequency of fusion, which is required. So what is done here? You take protoplasts from two different sources which you cannot cross or which cannot undergo sexual reproduction. So you have protoplasts. Okay, protoplasts from two different sources which otherwise cannot undergo sexual reproduction. Then they are made to fuse in vitro in a media, either through electrical means, through chemical means, and then you take regenerate the hybrid cell, hybrid cell, and this hybrid cell contain genome of both organisms, and it is very, very difficult to regenerate. I will come to those cases later. But then, conceptually, this is what happens in protoplast fusion. Okay, and this is just a big picture to show that how the two protoplasts are fusing. They come in contact with each other, and then the dissolution of membrane takes place at the union point, and then the hybrid cell is created. Now, when we, as I mentioned earlier, that when you have, for example, pools of two different cell types, this is cell A, cell B, and when they undergo fusion, so in that soup, you have these parental cells and also these different kinds of cells. So different kinds of combination of fused cell will be available. For example, you have cytoplasmic fusion. The nucleus could not go, only cytoplasm fused. So you have like this, a hybrid, we call them as hybrid. Wherever the nucleus has not gone into the hybrid, it's only the cytoplasmic hybridization, so we call them as hybrid. Parent A, parent B, and these are the hybrids where the nucleus also has moved in. So if you have nuclear fusion also taking place, which is the hybrid, and one is interested in, wherein you have the nuclear fusion, the one nucleus entering here. So you have the parental cell retaining its cytoplasm, only nucleus is entering into this, or vice versa. This going here or this going here, and you have these two hybrid cells. But the host cell retain its cytoplasmic ingredients. And in hybrids, you see this is the true hybrid where you have the cytoplasmic content of both the parents, from both the parents. Okay. So but then from this soup, it is you have to pick out these hybrid cells. Okay, and that's where the main problem was, that how to select a cell which has undergone nuclear fusion, and you have the true hybrid, what we call true hybrid. So then, based on the taxonomic relation, the kind of parents you take for protoplast fusion, so you have interspecific hybrids, meaning between different species of the same genus. You have interspecific hybrids or intergeneric hybrids. You have hybrids from two different genus, like potato and tomato. So the hybrid between potato and tomato was raised. Classical, very interesting, first experiment done with intergeneric plus fusion and regeneration of hybrid plant. And that plant name was given as tomato. Okay, and this plant, the idea was to generate a plant where you can have tomato in the shoot part and the potato in the root part. So, and then this hybrid could be generated, and they could have tomato in the shoot part and the potato in the root part. But unfortunately, these tomatoes never look like tomato, and the potato never looked like potato, and this plant could not be established in nature. Therefore, this work did not progress other than the scientific demonstration that it is possible to fuse two intergeneric protoplasts and raise a plant which looks like having the properties of both. You know, because in nature, if this kind of plant is to be exist, wherein you can have potato and tomato together on the same plant, it could have stabilized in nature itself. But since nature doesn't allow any such thing to happen, this could not be stabilized. And those products which were made by these plants were also not socially acceptable. So social acceptance is another part or issue which is involved into these kind of efforts when you are thinking of making newer combination, genetic combination, whether it is through genetic engineering or through such means. You know, whatever product you have, it should be socially accepted. Anyway, that is a different story and issue. We will talk about that later. But then what I'm trying to summarize here is that based on taxonomic relationship, you can have interspecific hybrids, intergeneric hybrids, and intertribal hybrids. And all of them have been tried in literature. If you see today, there will be examples where you can have this kind of demonstration, experimenting, products achieved, but then it just remained in literature as scientific query and nothing more than that. And this whole thing started in late 60s, early 70s, and continued into 70s. And after that, it died its own death. But then what it gave rise to that another concept that it is possible for two genomes to coexist in a cell and express. And that became the basis of genetic engineering. Because what in genetic engineering we do, we take gene from any source and try to put it in the host cell. This introducing gene can be from any source, bacterial gene, animal gene, plant gene, intergeneric, interspecific, intertribal, interphyla. You know, so you can take gene from any source and when it is moved into this cell, it expresses. I mean, it is different that it has to be made to express the gene by hooking it with appropriate promoter and terminator. But then these kind of the work, the protoplast fusion generation of hybrid demonstrated that it is possible to put two different genomes together in one cell, which expresses itself, you know, and gave rise to or it strengthened the concept of genetic engineering. Okay, so that is, you can say, is the positive contribution of protoplast fusion studies, you know. And then I have already talked based on contribution from two different genomes. You have symmetric, asymmetric, and these hybrids, you know. I'm not going into the details of it, and then selection. You know, because in absence of any biochemical markers, the visual markers were used to select the hybrid cell, like the herbicide resistance or the auxin autotrophy or chromosomal analysis, you know. These are some of the criteria which were successfully used as selectable markers, you know, or methods for selecting the hybrid cell. I mean, the advantage of somatic hybridization, as I had been mentioning, it is possible to bring together or create newer novel combination of gene pools, interspecific, intergeneric, you know, like tomato is the one, which was produced by hybridizing somatic cells between potato and tomato, and the production of fertile diploids and polyploids, transfer of genes uh, for disease resistance or variety of other traits it is possible. So these are some of the applications, and limitations are like any other protocol. They are very, very tough techniques to be followed and reproduced. So because of the poor regeneration of hybrid cells, I mean, the regeneration of hybrid cell is very, very tough, tougher than the single cell or unhybridized cells regeneration. Okay. And generally, the hybridized cell is non-viable. They generally die. So keeping it alive, and when the two genomes are interacting, in a hybrid cell, it is generally non-viable. Not successful in many plants, though as a concept has been tested in tomato and potato and tomato, where it could be possible variety of plants in literature. If you look through, there are many examples, but still there are equal number of plants where it could not be demonstrated. So with this, I come to an end of this beautiful application of plant tissue culture, wherein this is something new which was demonstrated, a possibility of bringing two different genomes together in a hybrid cell and making a newer combination. As I mentioned, though it did not succeed to give a plant which is producing two different products, like in case of tomato, but it definitely gave a positive information about that it is possible to bring foreign genes into the host cell where these genes can express themselves and give the trait to be regenerated. And the trait for which the A gene is responsible. And that became the basis of genetic engineering. Thank you very much for this, for listening.